Hello and welcome back to your fourth Essential Genetics video. And today we'll be talking about operons. This is what I meant by prokaryotic gene structure, but we're going to talk about operons anyway. But before we begin on, we have to talk about proteins first to understand this. Now, what is a protein? A protein is a polymer, just like DNA, or many other polymers you might have heard of, might have been polysaccharides. That's all I've got really. A polymer is a molecule that's made of building block molecules. The building block molecules of a DNA polymer are DNA nucleotides. Of a polysaccharide, it's the building block molecules or monomers are called monosaccharides. And for proteins, the building block molecules are called amino acids. The human body uses 23 different types of amino acids. It uses 23 amino acids. It uses 23 different types of amino acids to make its proteins. We produce 21 naturally, and we have to get the other two from our diet. Fun fact there. There's loads of proteins with various functions. Proteins can be used to carry things around the cell or around the body. They can be enzymes, which are structures that can start chemical reactions or accelerate the speed at which they happen. Proteins can also be structural. They can provide structural support for the cell. They can stop a cell from being squished to a certain extent or torn apart to a certain extent. Proteins can do a whole bunch of things. But to do their specific task, they need a very specific structure, a very specific amino acid composition. They need a certain sequence of amino acids, a certain amino acid recipe. But what determines what amino acids go into a certain protein? That would be the gene. What the gene is, it's a region of DNA which determines the amino acid recipe or composition of the protein. Now we can get into operons. Operons are multiple genes, more than one gene, or two more genes, that can be controlled by the same mechanism. They can be controlled as one unit. That's what an operon is. Here's just a quick overview of how a gene can be used to determine the amino acid composition of a protein. Essentially what happens is you've got your DNA region here, a region of a DNA molecule, and it's opened up. So and a copy of a region of the DNA of a certain sequence a certain copy of the region of that DNA molecule is made, but not as a DNA molecule, but as an RNA molecule. RNA and DNA are very similar. They have a few differences. Both are nucleic acids. Both of them are made of nucleotides. DNA is made of DNA nucleotides. RNA is made of RNA nucleotides. The sugar in a DNA nucleotide is deoxyribose, and the sugar in an RNA nucleotide is ribose. Both nucleic acids have nitrogenous bases. They share 3 of the same bases: adenine, thymine, and cytosine. Beg your pardon, I meant adenine, cytosine guanine. DNA has thymine, but instead of thymine, RNA has uracil. Thymine and uracil are very similar in structure, just a couple of atoms of difference. Oh, and RNA stands for ribonucleic acid. So an RNA copy of a DNA, an RNA copy of a DNA strand is made by RNA polymerase. So let's just zoom in here. We'll draw the RNA in white. So this is zoomed in here. And this RNA molecule is then taken to a structure in the cell called a ribosome, which then the ribosome will react with the RNA molecule, and certain amino acids will be added depending on the sequence of nitrogenous bases fed through the ribosome. Yeah, the ribosome again. Ribosome is something different entirely. I should also mention that this only occurs on one of the two strands on the DNA molecule. Operons are a major way in which genes are organized in prokaryotes. Prokaryotes are just cells without a nucleus. So cells without a nucleus. As opposed to eukaryotes whose DNA is kept in a nucleus. Prokaryotes are bacteria and archaea, for example. Anyway, enough of the background knowledge now. Let's get into how operons are actually structured. Oh, and one other thing. Operons do occur in eukaryotes, just not as often. They're just rarer in eukaryotes. Next video we'll get into how exactly protein synthesis plays out, but today we're just on about operons. So there's 4 parts to an operon. You have your premotor, you have your operator, and you have your genes. And of course, an operon is made of DNA, of course. Let's start with the promoter. The promoter is just sort of a landing site for RNA polymerase, the enzyme that will create the RNA copy of the genes. So RNA polymerase attaches to the promoter sequence. It'll bind to the nitrogenous bases, the sequence of nitrogenous bases, also known as the DNA sequence, called the promoter. Now next there's a barrier. Well, there can be a barrier here. There can be another protein here called a repressor. Now it has its own landing site, its own sequence, DNA sequence it can land onto, called the operator. When the repressor is occupying the operator, RNA polymerase physically can't get to the genes and can't make the RNA copy of the genes. So that's a problem for us, right? But when it's not on the operator, RNA polymerase can just go through and get access to the genes. So these are the genes here. So when it's not occupied, the RNA polymerase can just go right through and make its RNA molecule. Why go through all this at all? Remember how I mentioned that many genes can be regulated at once, or an operon is several genes that can be regulated as one or controlled as one. In a eukaryote, in protein synthesis or gene expression, RNA polymerase only really makes, most of the time, the RNA copy of the gene or the RNA transcript for one gene at a time. In an operon, which mostly occurs in prokaryotes again, will make the transcript for several genes at once. So you might have this might be gene 1, this might be gene 2, and this might be gene 3. And so when this RNA transcript is fed into a ribosome, then several genes will pop out. This RNA transcript is described as polycistronic, which is just a cistron is just an old word for gene. I'm going to give you an example of an operon that occurs in bacteria called a lac operon. Now imagine a situation where you've got some genes that break down lactose in the cell. So lactose, we'll just call them lactose genes. Genes in an operon tend to be related in function. So for example, you have all these genes that deal with breaking down lactose, which is a type of sugar. If RNA polymerase tries to get on the promoter, it'll be blocked by the operator. So how's it going to know? Because you've got a problem here. Because how's the RNA polymerase supposed to know? Because if you have all these genes in the cell being constantly made, there will be times where you don't need to make that certain type of protein. So you won't need to use these genes. How does it know when to use these and when to not? Turns out when there's no lactose in the cell, this repressor protein occupies the operator. But when there is lactose in the cell, what happens is a certain type of lactose attaches to the repressor protein, so lactose, And when it attaches to the repressor protein deforms and it gets off the operator. So it gets off the operator, and RNA polymerase can just make those genes. And it'll keep making those genes until there's no lactose left in the cell. And it knows this because the cell will just keep on making that repressor protein, and what will happen is there'll be no lactose there one day, and so there won't be lactose to deform the repressor, and so we go back to this, where the repressor occupies the operator, and the RNA polymerase can't get past it. But that's all for today. Hope you enjoyed. Next week, or next video, I should say next week, because we're making dangerous promises like that, we'll be discussing protein synthesis. Till then, goodbye.